right. It is 12 on the nose. Just in the interest of time, I think we should get started. Lindsay, do you want to introduce us and take us away?
Yep, I'm ready. Thanks, Michelle. Hello, everyone, and welcome to the first meeting of Ask the Researcher Mpa Monitoring Series. My name is Lindsay with the Ocean Protection Council, and I'll be facilitating this webinar alongside Steve Wirtz with the California Department of Fish and Wildlife, and AJ and Rochelle with Strategic Earth. This webinar is being recorded and closed captioned so we can make the discussion available to folks who are unable to attend live. If you have any questions or difficulties viewing the screen or calling in, please send a message to AJ either in the chat, through email, or by text. His contact information is posted in the chat. Because the large volume of participants, everyone will be muted until they're unmuted by the facilitator. During the Q&A, if you'd like to ask a question, please raise your hand and click accept when the message, "The host would like to unmute you," appears. During the Q&A, the facilitators will keep track of the hands raised and invite you to unmute when it is your turn. To help identify yourself and other participants, please go ahead and rename yourself on Zoom right now. To do so, you can click Participants in the meeting controls, locate your name, click More, and choose Rename. Please enter your full name first and last, and an identifier. If you'd like to enable closed captioning, please click CC in the meeting controls. We're pleased that you all could join us here today. It's so exciting to see all of you here, and we hope to see you with the following webinars as well. This series was designed as an opportunity for interested members of the public to interact directly with the researchers. The researchers are involved in California's MPA long-term monitoring projects, and in some cases in baseline monitoring as well. Our goal in facilitating this 8-part series is to support broad understanding of the research that is informing the MPA 2022 Decadal Management Review. Last fall, we hosted 4 community meetings to both inform the public about the Decadal Review and hear perspectives and priorities on MPA management. A consistent piece of feedback heard was to let folks hear directly from the researchers to help them better understand the science that is informing the review. These webinars are intended to be responsive to that feedback. But before we begin, I'd like to go over some meeting agreements that will help promote a safe and respectful meeting for everyone. So first, please stay muted when you're not speaking and try to minimize distracting actions. Please listen for understanding and acknowledge and seek clarification of others' perspectives and verify assumptions. Openly discuss issues with others who hold diverse views and approach discussions from a place of diversity and inclusion. Please keep comments concise and focused to the topics at hand. Personal attacks and offensive language will not be tolerated. And finally, please connect with the facilitators to talk through questions or concerns that you might have. We ask everyone to abide by these agreements. If you're unable to, we will invite you to leave today's meeting and watch the recording that will be posted in the coming days. You're also encouraged to email your input to the California Department of Fish and Wildlife at the address in the chat. Okay, we're very fortunate today to be joined by Dr. Mark Carr from UC Santa Cruz, who'll give a brief presentation on his work monitoring kelp forest ecosystems in the Marine Protected Area Network. After the presentation, we'll have time to hear questions from you all. And following Q&A, we'll hear from Steve with the department, who will provide a quick update on the Decadal Management Review. Now I'm happy to turn it over to Dr. Carr.
See, stop sharing. And how's that? Can everybody see this first slide okay?
Yep. Yes, I can see it.
Great.
You're good, Mark.
Great. Hey, thanks, Lindsay, and thanks for the opportunity to share some of the key summary points of the MPA evaluation studies that we've been conducting to inform the Decadal Review. And this is the only slide really where I'm going to acknowledge my 7 co-PIs on this project, as well as Barbara Spiker, who's a postdoc on the program at UC Santa Barbara, and Dan Malone, a researcher at UC Santa Cruz, both who have been instrumental in this work and the presentation, actually. So what I'm going to do is start with a brief summary of the background for the long-term monitoring program to provide all of you with some context for the design, the analyses, and the results that I'll be presenting. Then I'm going to touch on some of the overarching results. And those illustrate how species vary in their responses to the MPAs and how those responses vary across the biogeographic regions across the MPA network. And then I'll summarize with some general results and share some recommendations that we have that have been generated by the results of the project. For context, though, I wanted to quickly start with 3 ecological goals of the 6 overarching goals of the MLPA that motivate the kinds of questions and analyses that I'll be presenting. The first ecological goal, it was to protect the natural diversity and abundance of marine life, as well as the structure, function, and integrity of marine ecosystems. The second is to help sustain and conserve and protect marine life populations, including those of economic value, and rebuild those that are depleted. And then the third is to protect marine natural heritage, including protection of the representative as well as unique marine life habitats in California's waters. And certainly one of the iconic marine ecosystems and habitats in California's waters are kelp forests. But it's important to recognize that kelp forest ecosystems vary markedly along the coast of California with respect to the species composition that constitute the communities and ecosystems. So for example, up here in Northern California, you see that the predominant kelp that creates the forest is the bull kelp nereocystis, and the fish assemblage is dominated by rockfishes and some greenling species. As we move down to Central California, you see there's a mix of giant kelp and bull kelp that constitute the forest. And then still a large number of rockfishes, but a diversity of other species as well. And then eventually we get down into the Southern California region where it's predominantly giant kelp that's creating these forests. And the fish assemblage is a mix of some temperate as well as subtropical species, things like kelp bass and the California sheephead and Garibaldi that we don't see up in Central and Northern California. Because of that, to evaluate how these different species and communities respond to the establishment of the MPAs requires monitoring of MPAs with kelp across the entire state and MPA network. And as I'll describe in the next slide, this required creating a consortium of institutions that collaborated in the development of and implementation of the monitoring program. But I want to use this slide to point out, for example, that of Humboldt State, Reef Check, California, UCSC, UC Santa Barbara, and Occidental, there are the sites that they sampled are indicated by these different colored bullets. And you can see, for example, that Humboldt State and Reef Check did the lion's share of sampling in the North Coast, UC Santa Cruz and Reef Check along the Central Coast. And then down in the Southern Coast, there's UC Santa Barbara, the Vantuna Research Group at Occidental College, as well as Reef Check as well. Okie dokie, so these studies were all conducted using scuba surveys. And the earliest surveys started in 1999. But as the network and the number of MPAs grew over time, it required building up a broader team of investigators for the kelp forest work. And so we established this consortium that I just mentioned with UC Santa Barbara, UC Santa Cruz, the Vantuna Research Group at Occidental, Humboldt State University, and then in addition, the Citizen Science Program, Reef Check, California. We would monitor as many as 277 sites in a given year. And you'll see that The fewest sites on average in a given year was in the northern region. That increases in central California, and then it's greatest in the southern region, where the coastal area is largest and the number of MPAs is greatest as well. So it's important to recognize that the investigators didn't just make up whatever they thought would be important to study in these monitoring programs. We were given very important guidance by these two documents. The first is the Marine Protected Area Monitoring Action Plan, especially Appendix B, that really lays out nicely the questions that are related to the different goals of the Act. And then that was further developed by the Decadal Evaluation Working Group that was hosted again by the Ocean Protection Council and the California Ocean Science Trust. And so based on the kinds of questions, I want to start with clarifying our analytical approach for assessing population and community responses to the establishment of the protected areas, and how that includes three hypotheses. So this first hypothesis is that species targeted by fisheries should show a greater biomass increase over time inside the protected areas compared to those same species populations outside the protected areas. And I'll note that we use biomass because remember, it's the product of both the number and the size of the individuals that constitute a population. So the predicted pattern for that hypothesis is illustrated here, where we would predict that in the absence of fishing mortality, those species targeted by fisheries should increase greater inside the MPA compared to those same populations outside the MPA. The second hypothesis that we test is for those species that are not targeted by fisheries to exhibit similar changes over time inside and outside the protected area. And this is important because these species are not fished, their differences in their trajectories would be associated with differences in habitat. So they're a control for the habitat that we're monitoring in and out of the protected areas. And then finally, we'd also predict that a change in biomass of the targeted species over time should be greater than the non-species response over time, both within the MPA. So we're essentially asking, does this growing difference between the targeted species in and out of the MPA, is that a greater response than this change between the non-targeted species inside and out? And that's what's illustrated in this lower graph. So, but this is what it really looks like, right? So for every year, we go to multiple sites, we calculate a mean of, in this case, of the biomass of the targeted fishes within the MPA at this particular marine reserve. And then these vertical bars are the error around that estimate. And then we also show the error in our estimate of this relationship over time with this confidence interval in gray. So I'll be showing you some of these, and I wanted to make sure you were prepared for how to understand and interpret these trajectories, again, of the targeted species by fisheries and those that are not targeted by fisheries. But to do that means that we need to sample these populations both inside and outside of the marine protected areas. So I've illustrated here on the central coast of California, these two protected areas. This is the Point Lobos Marine Reserve, the Carmel Bay Marine Conservation Area. And I'll mention quickly here for background, remember that a marine reserve is a no-take marine reserve. Nothing is allowed to be taken. Whereas a conservation area does allow some form of take, right? So in this particular case, it allows all recreational take of finfish. And that's important because we can compare species responses based on those two levels of protection. But also, like I said, we have to monitor outside the protected areas so we can compare the responses inside and outside of those protected areas. And then what we do at each one of these little red dots or blue dots is a site where we conduct these transects, these 30-meter long by 2-meter wide by 2-meter tall transects. Those transects, replicate transects, are sampled at different depth levels across the width of the kelp forest. These are the many surveys that we do at a site for fish. And then we do a fewer number of benthic surveys to record the abundance of algae and invertebrates at these 3 different depths. Okie dokie. So we're blasting now into the first slide of results. And depicted here again, and let's start by focusing in right here on the south coast, if you will, right? Where again, we've got on the top graph, those species targeted by fishing. The lower graph are those species that are not targeted by fishing. Remember, the red line shows the trajectory of the targeted species within the protected area. The blue line shows the response or the change in abundance of the targeted species outside, and then the same for the non-targeted. So what you see going up here to the north coast, there's not much difference between the inside and outside of the protected area by those species targeted by fishing, as well as those that are not targeted by fishing. If we move to the central coast, those species targeted by fishing are starting to show differences, but they're not statistically distinguishable yet. And then we see absolutely no difference in and out of the MPAs of the non-targeted. But notice down here in the south coast and the northern Channel Islands, this is where we start to see these significant increases in biomass of the targeted species, right? And less evidence for differences of the non-targeted. But it's really down here in the northern Channel Islands where you see this difference in the response of the targeted species, no change in the non-targeted, and this change between inside and outside for the targeted is significantly different from that of the non-targeted. So there's no question that the response of the targeted species is attributable to the establishment of these protected areas. So we can also ask how these relative responses in biomass, inside versus outside, varies among the different species and among the biogeographic regions. So here I've listed, note the targeted group again and the non-targeted species collectively, but then a whole suite of other species individually. And I've done that for the North Coast, the Central, the South, and the Northern Channel Islands. And what is plotted along this axis is the relative change over time in the percent difference inside and outside of the protected areas. And so if a point lies to the left of no change, this is where there's actually been greater increases in the reference areas outside relative to the sites inside the protected area. In contrast, points over on the right side indicate where there's been a greater increase within the protected areas relative to the populations outside. And you can see I've given asterisks, as you'll see in the next slide, to those species that are not targeted by fishing. So there's 3 patterns in these results that are notable. The first one, note that as you proceed from the North Coast to the Central Coast and down to the South and Northern Channel Islands, you get this increasing proportion of species, including the targeted species, showing these positive responses within the MPAs relative to the reference areas, okay? The second is that although both the targeted, which is the very top bullet in each of these graphs, and the non-targeted, which is the second one, show positive responses. Notice how the targeted is always much substantially greater than the non-targeted species, right? Greater responses of those species that are being fished. The third is that in addition to the species as a whole, the targeted groups as a whole, certain species, especially in the southern coast, like California sheephead and the kelp bass, and then again down in the northern Channel Islands, California sheephead and kelp bass show marked positive responses to the establishment of the protected areas, right? And we know these are species that are heavily fished. With lower responses of all of these species with asterisks that are species that are not targeted by fisheries. And then we can ask the same for particularly important invertebrates and algae, comparing again the percent difference in the relative change over time inside versus outside of the protected areas. And here again, I've listed all of these different species of invertebrates and the bull kelp, the giant kelp, in each of the regions. And so the notable responses here, and notice that you don't see this as strong a gradient of increasing responses from north to south when it comes to the algae and invertebrates. But there's some interesting responses here. Look at this is red abalone showing a dramatic increase in the MPAs relative to outside, but you can't harvest red abalone on the Central Coast. So there must be some other factor that's contributing to, there's likely, I should say, some other factor that's contributing to that, maybe greater detection of the abalone in the MPAs over time. But then as you move into the South Coast, you see species like spiny lobster and the cucumbers showing strong positive responses. And again, spiny lobster out in the Northern Channel Islands. Again, species of invertebrates that receives pretty substantial harvest pressure. And then finally, notice that even in the North Coast, a positive, though not significant effect of bull kelp, positive effect of giant kelp in the Central Coast, significant in the South Coast, and in the Northern Channel Islands as well. So you see generally these positive responses of the kelps within the marine protected areas. Okie dokie. I now want to shift to thinking about how especially climate change, but especially the marine heat wave has affected species abundances and trajectories through time. And I'm going to do that with kelp because I think kelp's one of the most important species, right? It provides food and habitat to so many other species in the ecosystem. And I've plotted for the North Coast where bull kelp, remember, is the predominant kelp, the Central Coast and the South Coast, representative marine protected areas for each of those regions. So in the North Coast, this is Point Arena and Stewart's Point. And notice here in the North Coast that although bull kelp is quite dynamic, subsequent to the onset of the marine heat wave in 2014, bull kelp flatlines. And it does it in most of the MPAs that we look at. So it's a broad, and it does it also inside and outside of the protected areas across that region broadly. In the Central Coast, see that kelp both inside and outside of the protected area declined in say the Point Lobos Reserve, but it really did not change much in the Point Bouchon Reserve. So there's greater variation in the response of kelp to the heat wave in the Central Coast. And then when we get down to the South Coast, Point Vicente is representative there, and the Channel Islands, Skunk Point, representative there. You see that there's not a dramatic change in giant kelp subsequent to the marine heat wave, though notice that it declined pretty dramatically outside of the protected area here at the Skunk Point Reserve. But instead, notice that the percent in the increase in biomass of the kelps in the protected area is greater than outside of the protected area. And that's the case in both of these protected areas, right? Greater positive abundances and population trends within the protected areas. And this takes us to an interesting community level.
And Mark, just giving you a 5-minute check. Sorry to interrupt.
Yep, no, thanks, Rochelle. And so I've depicted that here for the Scorpion Marine Reserve out on the Channel Islands. At the very top up here, I've plotted the trend in the sunflower star, which is an important sea urchin predator. And in many parts of the California coast where we lost the sunflower star, purple urchins went nuts and kelp declined dramatically. And so you can see here how the sunflower populations declined dramatically post the sea star wasting disease in 2013. But interestingly, in this protected area, we see these positive increases in the California sheephead and the spiny lobster, both of which are alternative purple sea urchin predators. And then as a consequence, in this particular reserve, we see that purple urchin numbers are controlled, especially in the protected area where there's essentially 0 purple urchins in the presence of these increased abundance of urchin predators in that protected area. And then what we see as a consequence is this greater increase in kelp in the marine protected area relative to outside of the protected area, perhaps in response to this trophic cascade from these urchin predators to the abundance of kelp. So in summary, MPA effects clearly vary across the different species that we looked at. They also vary across the MPAs within and in between the regions, but they also vary especially across those biogeographic regions, the north, the central, the southern, and Channel Islands. They also vary in time periods, pre and post the marine heat wave. So the regional scales proved to be really the most insightful spatial scale to find these responses than, say, combining across the entire state. In general, the strongest population responses that I showed were in the South Coast MPAs and the Northern Channel Island MPAs, and the weakest were in the Northern California MPAs. The Northern Channel Islands and South Coast MPAs with the strongest responses have moderate to high fishing pressure outside of those MPAs. The Central Coast showed much more variable responses among species and MPAs. And in fact, the two southernmost MPAs, Point Sur and Point Bouchon, showed really the strongest responses in the Central Coast. And then, as I mentioned, the North Coast, there were no clear responses to the establishment of the protected areas. And this is in part because it's harder to get out and to collect the data to do these surveys. These are also the youngest MPAs and importantly, the shortest time series that we had to evaluate these effects. And then also the environmental disturbance, the marine heat wave caused really dramatic changes in kelp abundance that may have swamped any of the MPA responses. Those focal species that are heavily fished, the sheephead, the kelp bass, the spiny lobster, tended to show the greater responses to protection than other species. And so our overarching recommendations are, first of all, to continue this long-term monitoring of kelp forests and the environmental conditions for 2 reasons. One, I showed you that we get stronger abilities to evaluate the performance with longer time series. So we need as long of time series as we can generate, but also because these surveys are identifying important effects of things like the marine heat wave to these kelp forest communities. And so if we can continue on, we can continue to understand better the effects of climate change on this ecosystem. Another recommendation would be to prioritize further the MPAs that we monitor and evaluate. There's already been prioritization in this process by the department and the PIs, but it may be worthwhile to make a more cost-effective program to further prioritize and limit the MPAs. And then finally, as I showed you, the strongest responses were for those species that are fished strongly. And so that suggests that having information about fishing pressure around these MPAs at the reference sites and within the conservation areas that allow fishing could probably really help us interpret the various results that we're seeing across the different protected areas. And with that, I'll thank all of you for your time and attention, and we can open it up for questions.
Thank you, Dr. Carr. That was fantastic. It was a very helpful synthesis of your report. I can't take credit for that cheesy joke. That's AJ. But now we have about 20 minutes to answer questions from the audience. We ask that the questions be relevant to the topics at hand, specifically focused on the project that Dr. Carr just shared with us all, and that we leave space for everyone to have a chance to speak. You're invited to raise your hands either through Zoom or if you're on the phone by pressing *6, and one of the facilitators will unmute you when it's your turn. You are also welcome to ask questions in the chats, and we'll include those in the queue of questions for Dr. Carr. We'll read about one chat per person as time allows. We may not get to all of your questions today, so we strongly encourage you all to review the recording or follow up with any outstanding questions by sending an email to the Decadal Management Review email address. I already noticed a few questions in the chat, so maybe we'll just start off one here from Erica Yelensky. She asks, have you seen a change in species composition over the past 10 years? So Mark did touch on this a little bit and spoke about some notable species changes over time, but I'll add on, perhaps were there any unexpected species responses you observed as well?
You know, not so, certainly not in the North Coast, not very much in the Central Coast. And so if there were species changes, especially increases in abundance of the more subtropical species with warmer water, those would have been manifest in the southern region. And, you know, Jen can respond to this because that's where she works, but there has been, in fact, an increase in the abundances of some of those subtropical species.
Great, thanks. I see Melanie Moreno has her hand raised. I'm not sure if we want to unmute Melanie, and we'll go back to the queue in the chat.
Yeah, thank you. Hey, your data showed that there's a really significant benefit to kelp in areas that have more California sheephead. And I've noticed in my diving in southern Monterey Bay that post 2014, we had quite a huge influx of recruiting California sheephead. Mm-hmm. And those sheephead are just coming into changing to males. And so we have the potential of, you know, in keeping those California sheephead here to act as predators for urchins. Do you know if there is any move to try and protect those California sheephead from being overfished by both the spear fishermen and local hook and line fishermen?
So I'll quickly respond to that, and then someone from Fish and Wildlife may want to add on. But the important thing is that any of those sheephead that are in these no-take marine reserves are in fact protected, right? And so it'll be very interesting to track this through time to see if you see a relative increase in sheephead populations within the no-take protected areas compared to the conservation areas where they are allowed to be taken and areas outside of the protected areas. So see, this is cool. This is how you can, and a great observation on your part, Melanie, and it shows how you can use the protected areas almost as a long-term experiment to see the longer term consequences of protection of some of these functionally important species in the system.
Can I ask, I noticed in southern Monterey Bay, there's kind of a patchwork of SMRs and SCMAs, and that means that they're so close together. I don't know how far sheephead tend to roam, but I'm guessing that they're kind of roaming back and forth. Does that make a difference?
Well, we'll have to see, right? But remember that we tried to design the individual protected areas to be large enough to accommodate the home range movement of many of these species. So we'll have to see whether the ones that you're referring to, whether that the Julia Pfeiffer Marine Reserve was of sufficient size, or whether there's substantive spillover into the Ed Ricketts and the Pacific Gardens conservation areas.
Thank you.
You bet. Thank you. Good question.
Thanks, Melanie, for that question. Why don't we pop over to one in the chat, and then I see Wendy has her hand raised after. We have a question from, I'm not sure if it's Jan or Jan Roletto. Have you seen a change in species comfort? Oh, I'm sorry, we already did that one. Sorry, let's skip to the next one after that. Karen asks, what are the delineations between North Coast, Central Coast, and South Coast? A quick clarifying question there.
Yeah, well, and also what I should point out, you have no idea how many discussions and arguments were behind that decision for the kelp forest people alone, right? Because we wanted to delineate, so separate from the delineations that the department uses for the northern and central and southern, we wanted to ensure that the delineations we made were actually based on differences in species composition, especially of the fishes, but of the whole community actually within the kelp forest. And so that's one reason why we pulled out the Northern Channel Islands as a really distinct biogeographic region from the rest of the South Coast area. So that's one important delineation. The South Coast delineation, as you can imagine, is at Point Conception, right? North of Santa Barbara and south of Point Bouchon. And then the delineation that we used between Central California and Northern California is around the San Francisco Bay area. But remember, we don't monitor kelp forests north of southern Monterey Bay. As you go further, and there's two reasons. One, there aren't many protected areas with kelp north of southern Monterey Bay, but what there are sharks. And so we don't have data for any of the MPAs north of Monterey Bay, yet we make that delineation right around the Golden Gate, essentially, anywhere between there, between Monterey Bay and the Golden Gate.
Thanks, Mark. And maybe as a quick follow-up, you already did touch on some of the difference between the results between the different regions, but perhaps maybe thinking on a couple species in particular, and maybe more oceanographically, why do you think the results were so different between North, Central, and South?
Yeah, and you know, our impression is that the primary driver of responses is the relative fishing pressure, right, outside of the protected area versus within the MPA. And you know, if you look at the literature on marine reserves and protected areas in general, that's a kind of a global phenomenon, right? That obviously the greater the fishing mortality outside and the level of successful protection inside is really going to drive those differences in the populations. And so our feeling is that, our impression is that there is stronger fishing pressure in the Southern California region relative to Central California and Northern California, but especially for certain species. Like I said, for California sheephead, the kelp bass are only taken recreationally, not commercially, because they are such a favored recreational finfish, right? They're hammered in southern, I mean, because it's so popular. I shouldn't say they're hammered in the sense that they're being depleted, but they're certainly sought after. And then the California sheephead is not only taken recreationally, but commercially as well. So that's what is largely driving those regional differences, is the species that are being experiencing heavy fishing.
Excellent. I'm glad you touched on that a little bit more in depth. And there's similar responses we've seen with our other monitoring programs that you'll hear about in the coming webinars as well. Wendy's been so patient with her hand up, so we'll go to Wendy next.
Hi, thank you so much for this opportunity to speak to you about this. My question was about the graphic that you showed with the different kelp populations and densities. In the southern region, it looked like there was a fairly steep and then sustained decline in the giant kelp from like the 90s.
Oh yeah, look at
To the
right.
2000s. I'm wondering if you know anything about what caused that. It was
Yeah.
further down. Oh,
Right. Yeah, you were
Thank
looking at it. But am I still sharing my screen?
Hi, Mark, I am, but you're welcome to share your screen.
Oh, I'm sorry. Oh,
but
okay.
she is showing the slide I was talking
Oh,
about.
good. No, okay, great, great. No, this is good. But I have to move a chat so I can see.
Yeah.
Yeah. Hey, great. No, so I can't I can't put my cursor on there, but Lindsay, if you can see down in Point Vicente from 1990 to around 2007, there's a dramatic decline in the overall abundance of kelp. I don't know what that is. I don't know necessarily what was driving that, because it's all post one of the stronger El Nino events, right? Of like 82, 83. You see that kelp rebounded in that earlier period, but then it declined and stayed low until around 2007. But having said that, if you look at unfortunately I didn't plot it on here. If you look at the general water temperatures over this period and the magnitude and frequency of upwelling along the coast, those waters were warmer over that period than in some of the other periods of that time series. So it may again, and it probably was related to essentially the ocean climate, the ocean temperatures over that period that we know affect the productivity of the kelp plants.
Right. Okay, thank you. I know that was before your sampling period, but I figured you would know a little bit about it. So I appreciate that. Thanks.
Thanks, Wendy. We have another question in the chat here from Jacqueline. Have you seen a correlation between giant kelp recovery in the South Coast region and where previous kelp seeding efforts were implemented? And/or is it primarily attributed to the additional urchin predators? This is a long one. Also, is giant kelp more resilient to warming ocean temperatures?
Okay, there's about 40 questions there, so we'll have to kind of dive a little bit. But the first one is that a lot of the seeding activities are not done within the protected areas, and they're done in very specific locations that don't necessarily correspond with where we were doing our surveys for the protected area evaluation. So because of that spatial mismatch of where we sample versus where the seeding is done, I can't really tell you, but they probably don't contribute very much to the responses that we're seeing here. Jen Cassell, who has done the surveys down in that region, could maybe elaborate on that. but the other is, sorry, another question was whether the greater increases in the protected area was largely due to differences in the urchin populations, I believe, right?
Yep.
And that is the case in particular MPAs, especially out in the Channel Islands. Yeah.
Mark, do you want me to take this? This is Jen.
Sure.
Okay, yeah. So, Jacqueline, first off, yeah, Mark is correct. Even the long history of small scale kelp restoration projects that have happened in Southern California, they've been very, very small efforts that have gone for limited amounts of time for the most part. I'm talking about all the way back to the 60s. So our overarching MPA monitoring across all of Southern California would not be at the scale where we could detect effects of those specific and small spatial scale projects. But hopefully all of those are being monitored on their own for effectiveness. And so there should be, you know, the folks who do that kind of restoration work should be able to say something about their areas. When we look at all of Southern California and the Northern Channel Islands together, then as Mark indicated, we really think right now that Southern California, two things are happening. One, we have a very long history of very extreme dynamics between urchins and kelp over history. They have come and gone. We've had Barren formation and then kelp coming back, more so in fact than other parts of the state. So it's a natural cycle to some extent. And then secondly, we do have those additional urchin predators, the California sheephead and the lobster, which Northern California currently does not have. And the last part of your question, is giant kelp more resilient to warming ocean temperatures? That is a great question. And there's a lot of work being done on that right now throughout California and also globally to investigate whether there are strains of kelp, giant kelp, that may be genetically more resilient or more adaptive to warming temperatures, especially if they come from places that experience warm temperatures. So the jury is still out on that in California, but there's a lot of really interesting research taking place on that exact question right now. If we have strains that are more resilient, potentially they could act as sources for restoration in other areas. So thanks, Mark.
If I can, I'd like to use this question to raise an important caveat when we interpret some of the results. So for example, I've mentioned here, and especially in Kyle Kavanaugh's analyses using Landsat to track the recovery of kelp forests from the marine heat wave inside and outside protected areas, he sees a much more rapid and greater recovery in protected areas compared to outside of the MPAs. But there's an important caveat to that, okay? When we were designing these protected areas, when it came to determining whether the protected area really contributed to the protection of kelp forests, we would identify kelp forests that were large and stable through time, and then those would be protected or targeted for protection by the MPA. So there's potentially an inherent bias for kelp to be more resilient within the protected areas, because when we were creating the MPAs, we were going after those really large and stable kelp forests. So that's something that we'll have to explore in greater detail, but I just wanted to put that out. And this is fundamentally different from what Jen was just describing, which is this effect of higher level predators on urchins in the protected areas, contributing to the recovery of the forests as well. Okie dokie.
Thanks, Mark and Jen. That was super helpful. And I'm glad you mentioned the different types of methodologies used for this project. It's one of the really cool things. And in addition to the in-water monitoring, there's the Landsat, the satellite-derived data that's being brought in as well. I just want to note that we are lucky to have department staff on the call too, and they are chiming in with some answers in the chat as well. I saw Steve already wrote something about sheephead management there. So that's really great. They're able to reply in live time. Eric has his hand up, so let's move to him next. If we unmute Eric.
Hi, this is Eric Brasky from Laguna Beach. Thank you for the excellent presentation. I'm just wondering, so I heard you discuss differences in fishing pressure between the various geographical regions.
Yeah.
Is it also possible that home range is playing a role? I know that sheephead are very territorial, and maybe that's also part of the reason why we're seeing such significant gains down in the South Coast region.
Yeah, excellent question, right? What Eric is suggesting is that because the species in Southern California have a much smaller home range, they may get greater protection over their lifetime within an MPA compared to species to the north. I think there's some to that, Eric, actually. But it's interesting, on the other hand, there's a lot of the rockfishes in Central California that likewise have very limited home ranges. In fact, Jan Freiwald, who's the head of the Reef Check California program, did his doctoral dissertation studying and summarizing home range patterns for reef fishes along the coast of California. And even up here, many of the reef fish species have home ranges that are well within the spatial area of the protected areas. So it may be a contributing factor, like you're saying. I don't think it explains all of the patterns. I think that the differences in fishing pressure are probably stronger than the home range differences. But again, a great research topic.
Lots of great questions. Oh, sorry about that. Lots of great questions so far. Just a quick reminder, we have about 3 minutes left for questions. So if you have a burning one, go ahead and raise your hand or put it in the chat. We have one that Jeff wrote already. He's talking about sea urchin barrens increasing over time. And he ends with, how can the goals of the MLPA be upheld without direct intervention to countervail perpetual barren dead zones? This is a tough question, so appreciate your opinion here, Mark.
Well, this might be one to throw to Fish and Wildlife. If the question is about is it reasonable? Should we be thinking about intervention within the protected areas to reduce the number of urchins in those areas to enhance or facilitate the recovery of kelp in the protected areas? And there, it's a complicated question. And we're going to learn a lot from what we're seeing in the North Coast and the Central Coast with respect to the patterns of forest recovery inside and outside of the protected areas and related to urchin densities. It's interesting, others can maybe speak to this, but one of the most shocking observations that I have been told of on the North Coast is that in some of these areas where they've seen bull kelp recovering, there's lots of purple urchins. I would have never, okay, this is where Mark's going to bear his soul, saying that I would have never predicted that. I would have been one of the first ones, and I've said it in many newspaper articles, that you've got to get rid of these purple urchins if you ever want a recovery of a forest up there. But those observations prove me wrong in a big way. And so it's going to be really interesting to see, whether we can see some of this kelp recovery that is maybe more related to the oceanographic conditions, right? Cold, nutrient-rich water, as opposed to the abundances of the sea urchins in facilitating that recovery. So it's a complicated question. And if Steve or Becky or someone wants to comment on allowing urchin removal within protected areas, they could do so.
Yeah, Mark, thanks. It is very complicated, and kind of some of the present part of Mark's presentation, he showed in Southern California where we had some of the oldest MPAs out at the Channel Islands, and you see the natural predators grazing down the purple urchins. And so we really have to think hard about manipulating these environments, because we do consider them natural laboratories. And we're hopeful as they, quote, naturalize, that you wouldn't have to do this type of manipulation. So we are working with our partners at the Ocean Protection Council to kind of figure out how we may or may not allow that type of work to occur. And all the work that's being generated, as Mark had mentioned a moment ago, through this decadal management review will help us with that decision process. Because it's, it's an emotional interest and concern of the public when they see like loss of the kelp forests. But we do consider these natural laboratories, and we'd like to see them recover on their own if they can.
Yeah, if I can follow on Steve's comment, he was spot on that those trophic cascades that Jen has shown in her research, and I showed you in this presentation, if we had allowed people to go in and remove those urchins in those protected areas, we would have never been able to determine whether the decline in urchins was due to a natural process or the human intervention, which I think would be a huge loss, right? You can always possibly do the human interventions outside of the protected areas and then compare the results of the intervention versus the natural mechanisms of recovery, which will be, really fun to follow into the future.
Thanks, Mark and Steve. I know that's, as you've already pointed out, a very complex question. So I think you guys did a great job kind of explaining where we are today and what we've seen so far. With that, we need to shift gears a little bit. So thank you all for the great questions. As we stated up top, if you have any additional ones, please feel free to email that Mpa Management email address. We'll share it again later in the presentation as well. But now I'll turn it over to Steve, who'll give us a brief update on the Decadal Management Review.
Great. Great. Just once again, I want to reiterate, it was a fantastic presentation, and I always learn listening to the talks that Mark gives, and those are some awesome, insightful questions. We really appreciate those. I want to just take a couple minutes really quick, kind of highlight why we're doing this decadal management review. As you may know, the department completed planning of the network in 2012, and you can go back if you want. And so following that, we updated the master plan for marine protected areas and established a 10-year management review cycle, which we're in right now in the review process, and established the Mpa Management Program, which is composed of 4 pillars, which you may be familiar with. This is outreach and education, enforcement compliance, research and monitoring, and policy and permitting. That's all being looked at in this review. And if you could advance, Lindsay, that'd be great. So we're pulling all this information together for each of those pillars of the program for a report to the commission that'll be available for the public review in January of 2023, and it'll be formally transmitted to the Fish and Game Commission. It'll include the information you heard today from Mark, the research and monitoring. It'll include baseline and long-term monitoring data for the 8 projects that you'll be hearing about over the next couple of weeks. So stay dialed into that if you're interested into the research. We've been working with California Native American tribes to better understand traditional ecological knowledge and their perceptions of MPAs to help inform our management. We've also been meeting over the last year and a half with stakeholders like yourselves, agency personnel, government organizations, NGOs, to receive feedback to help us generate this report to identify, the challenges, accomplishments, and gaps in our knowledge. In addition, the department has been working with our fishery scientists and our law enforcement division to work up the data that we have in-house on that type of information. And then we were fortunate, can you click we were fortunate to have the Ocean Protection Council fund 2 independent science teams. One looked at performance objectives so we can effectively and objectively review the performance of the network. So we're very fortunate to have that. Mark and Jen were part of that process. And there was also a team that looked at the potential for the MPAs to have resilience to climate change. All of this information will eventually result in gaps in our knowledge, additional questions, which was awesome because that's how you learn. We're hopeful that it will result in refined, cost-effective management strategies and adaptive management recommendations that the department can give the commission to consider. And that'll take place in 2023, like I mentioned. We go to the next one real quick. I know we're right out of time. So in addition, we have a landing page on our department's webpage. If you want to stay dialed in and informed, I recommend you check this out. We'll paste the link in the chat so you have it. But this is the best way to stay informed of the things that are happening as we move forward to 2023. Just real briefly, in 2023, after the commission meeting, there'll be a subgroup of the commission's commissioners that will dig deeper into these adaptive management recommendations, and there'll be a public symposium where you can be engaged. So all that will be posted to this webpage. So I recommend just checking it out. And at the bottom, we have our Mpa Management Review Wildlife email address. And so if you have any questions about the review, send them to us. My staff and I try to respond within 48 hours. And it doesn't have to be the review. It can be anything about MPAs. We're always excited to answer those questions. So I know that was speedy, but if you didn't catch it all, please just send us an email. We look forward to hearing from y'all. And just once again from the department, we really appreciate the presenters and the nice comments that came in. Thank you.
Thank you, Steve, and thank you all so much for being here today. And a huge thanks to Dr. Mark Carr and Dr. Jen Cassell and their team for being a part of this and taking their time to pull this all together. I hope that these types of informal discussions will help make the science informing the Decadal Review accessible to all those interested. I know we're running over a little bit here, but I'll share a couple more things with you all. As a reminder, the full long-term monitoring reports, these amazing technical reports each of the researchers have compiled are available on California Sea Grant's website. That link's being dropped in the chat right now. As Steve said, always go to the department's landing page for the Decadal Review. That's already been shared. This meeting and all future webinars are recorded, and those will be posted on OPC's webpage and linked to that same CDFW Decadal Review page as well. And we're excited to say the next webinar is this Thursday. So that one will be featuring Dr. Pete Ramundi talking about his work in the Rocky Intertidal. So we hope that you tune back in this Thursday from 4 to 5. Again, if you have any additional questions or comments that we didn't get to today, please email the department. And thank you all again, and hope you have a great rest of your day.